So at Vincere Bio, what we're doing is basically repairing mitochondria, which are the powerhouses of your cells. And in that, we're looking to stop Parkinson's disease and additional disease indications that have to do with aging. The hypothesis basically is that your mitochondria get damaged, just it's life, right? They get damaged all the time, and there are machineries in the cell that remove these damaged mitochondria, but they become less efficient as we age. And this deficiency in being able to remove the damaged mitochondria, now you have these leaky power plants hanging around the cell. They can cause aging. There's a very nice reciprocal relationship there where actually if you can enhance mitophagy, fruit flies, for example, can live 30% longer and they have better health span. The females are fertile for a longer period of time. So we know there's a very direct bidirectional relationship with aging. Neurodegeneration, if you have mutations in this pathway, you get early onset forms of Parkinson's disease. And then, of course, immune and inflammatory responses, because mitochondria are essentially like little bacteria with their own DNA. So if they lyse, they get recognized by your immune system. So the solution is actually quite simple. The cell already has a mitophagy mechanism in place, and our molecules enhance this process by inhibiting USP30, which I'll tell you about, and this returns the cells back to a healthy state. So this is what our pipeline looks like. We're looking at multiple indications for this panel. Of course, Parkinson's disease is the relevant one. We have completed non-GLP tox studies and are now doing GLP tox studies. So we'll be looking to be in the clinic in 2026, later part of 26. We've also partnered our skin aging molecules that are for topical applications with Beiersdorf, which is the parent company of Nivea and many other brands you probably know about. And then we have some peripherally restricted molecules that we're taking forward for cardiorenal indications as well. So the way these molecules work, I mentioned that they inhibit an enzyme called USP30. USP30 is right on the mitochondrial membrane, and it basically takes ubiquitin tags off of the mitochondria. These ubiquitin tags are the signal to the cell to come and remove the mitochondria. So if they are taken off, then the cell doesn't recognize the damaged mitochondria and will not remove it. So by inhibiting this, we are able to actually get the cell to recognize the damaged mitochondria. And the nice part of this is that it doesn't really do anything to healthy mitochondria. We just see no effect on healthy mitochondria with this molecule. So this is work by others. This is from David Simon's lab at Harvard that was recently published showing neuroprotection in an animal model. Again, we know that mutations in mitophagy cause Parkinson's disease, so the cause and effect relationship is already in humans. Then looking at animal models, which are not perfect for Parkinson's disease or any other neurodegenerative disease, right? But we do see this neuroprotection in this synuclein model. We see a reduction in phosphorylated abnormal forms of alpha-synuclein and a benefit in motor deficits as well. So the next step is then to translate this to humans by making an orally bioavailable molecule that can be taken. And that's basically what we've done. So this is one of our molecules in human neural Ren cells. On the y-axis is a measure of mitophagy. On the x-axis, increasing concentration of the molecule. If you give it when the mitochondria are healthy, we don't see anything. There's no increase in mitophagy. If you damage the mitochondria with a mitochondrial toxin like CCCP, we now see an increase in mitophagy in a dose-dependent manner, and the molecules are quite potent in the low nanomolar range. Then in vivo, we can utilize mice that have a fluorescent tag on the mitochondria that changes color when it goes into the acidic environment of the lysosome. So in young mice, you can nicely see a robust increase in mitophagy in the dopaminergic neurons, which are the neurons we're interested in the brain. Now, we already know, as I mentioned, that there's a relationship with aging. So big question is, can we do this in aged animals or aged people who we know have a deficit in this pathway? So we tested it in old animals, and this, as you can imagine, 14 to 20-month-old mice, took some time to age these transgenic mice. This is equivalent to 56-year-old humans, normal age for Parkinson's patients. And what you see is, what's been reported in literature, we see as well that the mitophagy is lower in these older animals, but we're able to very robustly increase the mitophagy. So not just back to young levels, but young levels with the mitophagy enhancement. So we're very excited to see this. We have a development candidate that I won't get into, but we can disclosed under CDA that has all of the nice properties. It's potent, it's selective, it's oral, brain penetrant, and it increases mitophagy only when mitochondria are damaged. No toxicity that we can see up to 14-day rat and 10-day dog studies that we've conducted thus far. And then I just want to mention also that I'm a neuroscientist also. Everybody here is probably interested in neuro. And I used to say, if it's from here down, don't talk to me about it. But the data tells us some interesting stuff. So kidneys, for example, are another energetically demanding organ. And if you look at multiple different models of kidney injury, so here we have ischemic reperfusion, IRI, rhabdomyolysis, or cisplatin, which is a chemotherapy agent. All of these cause kidney injury. And if you give our molecule even 24 hours after the kidney injury has been initiated, we're able to see a reduction in kidney injury. So again, this suggests that this is not limited, the benefit is not limited to the brain, but any energetically demanding tissue where you have damage to the mitochondria causing damage to the tissue. And then this was a collaboration with a group at University of Michigan with abdominal aortic aneurysm. You can see the aneurysms right here in the abdominal portion of the aorta, as the name indicates. And if you give USP30 inhibition, you're able to lower the incidence from about 2/3 to 1/3, so quite significantly, and the size of these. And there's also a reduction in the immune markers here. You're seeing sting. So again, this is a systemic kind of benefit when mitochondria are damaged. And then this is the Beiersdorf collaboration, which now they have the molecules, they're the ones taking it forward, but there was a publication already, not ours, and this was not our idea. They're quite well-versed in mitochondria as well, but you can see a reduction in inflammatory markers and an increase in collagen production. So hopefully that will make it to the clinic faster than the CNS molecules that will have to go through much, much more scrutiny. So this is where we are. This is the timeline. We're looking to partner these either at IND stage or after phase 1 clinical trials. We're planning to take them through planning at this point to take them through phase 1 unless they get acquired before that. So here we're giving you an update on everything up to the GLP tox, and then in April we'll have the GLP tox portion done. So if there are any pharmas that we haven't talked to, which I think we've talked to pretty much everybody, we'd like to get everybody under CDA by April so we can really hit the ground running on diligence. And with that, we have a fantastic team. Everybody here has done several approvals. We have a fantastic SAB, and a big thanks to our funders, especially, you see some big names up there. And also I'll give a special shout out to the Michael J. Fox Foundation and NIH for the non-dilutive funding as well.